Good day everyone, welcome to SlideMed. Today we're going to be discussing on erythropoiesis, which is the second video of our red blood cell series. Our first video is also on YouTube, okay, so you can check it out, okay. So this is going to be our second video, erythropoiesis, and under erythropoiesis we're going to look at the definition of erythropoiesis. We're going to look at different sites where erythropoiesis can be carried out. And we're also going to look at the process of erythropoiesis. We want to look at how it develops from the stem cell level. We want to look at the changes that occur during erythropoiesis, the changes that occur on the cell, and then also look at the stages, different stages of erythropoiesis. Then also, we're also going to look at the factors necessary for erythropoiesis to take place. So erythropoiesis is the process of the origin, development, and maturation of erythrocytes. Okay, process of the origin, development, and maturation of erythrocytes. While hematopoiesis or hemopoiesis is the process of origin, development, and maturation of all the blood cells. Okay, erythropoiesis is just for erythrocytes, while hemopoiesis is for all the blood cells. Now let's look at sites of erythropoiesis. In fetal life, erythropoiesis occurs in 3 stages. The first stage is the mesoblastic stage, the second stage is the hepatic stage, and the third stage is the myeloid stage. The mesoblastic stage, during the first 2 months of intrauterine life, the RBCs are produced for the mesenchyme of the yolk sac. That's the mesoblastic stage, as it occurs during the first 2 months of intrauterine life. So RBCs are produced from the mesenchyme of the yolk sac. While the second stage, the hepatic stage, occurs from the 3rd month of intrauterine life. At this stage, the liver is the main organ that produces red blood cells. The spleen and lymphoid organs are also involved. The 3rd stage is the myeloid stage. It occurs during the last 3 months of intrauterine life. The RBCs are produced from the red bone marrow and the liver. So remember, 3 stages: mesoblastic stage, hepatic stage, myeloid stage, all within fetal life. Let's move on. In newborn babies, children, and adults, the site of erythropoiesis. In newborn babies, RBCs are produced from only the bone marrow. Okay, from only the bone marrow. Then, up to the age of 20 years, RBCs are produced from the red bone marrow of all bones, long bones, and the flat bones, mostly from the long bones and from the flat bones. Then, after the age of 20 years, RBCs are produced from membranous bones like the vertebra, sternum, scapula, and the end of long bones. Okay, after 20 years of age, the bone marrow of the shaft of long bones becomes yellowish because of fat deposition and loses its erythropoietic function. Now it's at this stage that the liver and spleen may now produce blood cells if the bone marrow is destroyed or fibrosized because of fat deposition. Okay, so let's move on. Let's look at the process of erythropoiesis. Now, erythropoiesis doesn't just happen like that. There are cells that introduces the production of erythropoiesis, and they're called stem cells. Stem cells are primary cells capable of self-renewal and differentiating into specialized cells. Hemopoietic stem cells are the primitive cells in the bone marrow which give rise to the blood cells. Hemopoietic stem cells in the bone marrow are called uncommitted pluripotent hemopoietic stem cells. They are uncommitted because they can give rise to any blood cell. They can give rise to anything. Okay, so they are uncommitted. They are not committed to something. Okay, pluripotent hemopoietic stem cells is a cell that can give rise to all types of blood cells. In early stages, the PHSC are not designed to form a particular type of blood cell, and it is also not possible to determine the blood cell to be developed from these cells. Hence the name uncommitted pluripotent hematopoietic stem cells. Okay, so when these cells are designed to form a particular type of blood cell, the uncommitted PHSCs are called committed PHSC because now they're no more restricted. They're now designed to form a particular type of blood cell. So these cells are restricted to give rise to one group of blood cells. Okay, so let's break them down. Committed pluripotent hematopoietic stem cells are of 2 types. We have the lymphoid stem cells and the colony-forming blastocytes. The lymphoid stem cells and the colony-forming blastocytes. The lymphoid stem cells give rise to lymphocytes and natural killer cells, while the colony-forming blastocytes give rise to myeloid cells. When grown in cultures, these cells form colonies, hence the name colony-forming blastocytes. Now we have different units of colony-forming cells. There are colony-forming unit erythrocytes, which is the colony-forming blastocyte for red blood cells. We also have colony-forming unit granulocyte monocytes, which helps in producing the cells responsible for the defense mechanism. Like example, the granulocytes, we have the neutrophils, we have the basophils, we have the eosinophils, and we also have the monocytes, okay, which is also a cell for defense mechanism. Now we also have colony forming unit megakaryocytes. This is majorly for the platelets. Okay, so let's understand that. First of all, lymphoid stem cells, colony forming blastocytes. Then under colony forming, we have colony forming unit erythrocyte, colony forming unit granulocyte monocyte, and colony forming unit megakaryocytes. So we can see the diagram here. Pluripotent hematopoietic stem cell. I'm going to leave it here for some time so you can study it. You can see the differentiation. We can see the colony forming blastocyte and the lymphoid stem cell. Lymphoid stem cell go lymphoblast, lymphocyte, then colony forming erythrocyte. You see the stages of erythropoiesis there. So maybe you can rewind, then try and study this diagram. Okay, let's move on. Now let's look at the changes during erythropoiesis. We see that cells of colony forming unit erythrocytes pass through different stages and finally become matured red blood cells. Now, during these stages, 4 important changes are noticed. Number 1 is the reduction in the size of the cell. The reduction in the size of the cell, it reduces from the diameter of 25 to 7.2 micrometers. Okay. Number 2 is the disappearance of nucleoli and nucleus. Number 3 is the appearance of hemoglobin. And number 4 is the change in staining properties of the cytoplasm. So let's take note of these important changes. Okay. Take note of these important 4 changes. Reduction in the size of the cell, disappearance of nucleoli and nucleus, appearance of hemoglobin, and change in staining properties of the cytoplasm. Now let's look at the stages of erythropoiesis. There are various stages between colony forming unit erythrocyte cells and matured red blood cells. Number 1 is PROERYTHROBLAST. Number 2 is early NORMOBLAST. Number 3 is intermediate NORMOBLAST. Number 4 is late NORMOBLAST. Number 5 is reticulocyte, and number 6, which is the final stage, is matured erythrocyte. Matured erythrocyte. Now, the PROERYTHROBLAST is the first cell derived from colony forming unit erythrocytes. It is very large in size with a diameter of about 20 micrometers. Its nucleus is large and occupies the cell almost completely. PROERYTHROBLAST does not contain hemoglobin. The cytoplasm is basophilic in nature. Now let's take note, there's something that happens in this stage. One important thing, synthesis of hemoglobin starts in this stage. The matured red blood cell contains hemoglobin. So it's at this stage that hemoglobin is being synthesized at first. So synthesis of hemoglobin starts in this stage. The PROERYTHROBLAST multiplies several times and finally forms the next cell stage called early NORMOBLAST. Early NORMOBLAST. So let's move on to early NORMOBLAST. The early NORMOBLAST is smaller than the PROERYTHROBLAST stage with a diameter of about 15 micrometers. Now it's at this stage that the nucleoli disappear. Okay, not the nucleus. The nucleoli disappear at this stage, early NORMOBLAST. The cytoplasm is basophilic in nature also. Therefore, it can also be called basophilic NORMOBLAST. Basophilic NORMOBLAST. That's because of its basophilic nature. It can also be called basophilic NORMOBLAST. This cell develops into the next cell stage called intermediate NORMOBLAST. So let's write on. In intermediate NORMOBLAST, the cell is smaller than the early NORMOBLAST with a diameter of about 10 to 12 micrometers. The nucleus is still present. Now, something very important happens at this stage. Hemoglobin starts appearing. In the first stage, the synthesis of hemoglobin starts, but it's at this stage that hemoglobin starts appearing at the stage of intermediate NORMOBLAST. The cytoplasm is basophilic in nature, but because of the presence of hemoglobin, which starts to appear, it stains both acidic as well as basic stains. So this cell is also called polychromatic erythroblast. Polychromatic erythroblast. Because of its diverse staining abilities. It stains both acidic as well as basic. So it's also called polychromatic erythroblast. This cell also develops into the next cell stage called late normoblast. Late normoblast. Now, in late normoblast, the diameter of the cell decreases further to about 8 to 10 micrometers. The nucleus becomes very small with a very much condensed chromatin network. So the nucleus starts degrading, it becomes very small with a very much condensed chromatin network. The quantity of hemoglobin increases and the cytoplasm becomes almost acidophilic. Okay, it becomes almost acidophilic because of the quantity of hemoglobin that is increasing. Now, at the final phase of late normoblast, the nucleus disintegrates and disappears. That's the special thing in late normoblast. But note, it is at the final phase of this late normoblast, the nucleus disintegrates and disappears, and that phenomenon is also called pycnosis. You can look it up. Pycnosis. Okay, it's a phenomenon that characterizes the disintegration and disappearance of the nucleus at the final phase of late normoblast. Then the late normoblast develops into the next cell stage called the reticulocyte stage. Now, at this stage, reticulocyte is also the reticulocyte is also known as the immature RBC. It's not matured yet. Okay, it's also known as the immature RBC. It is slightly larger than mature RBC. The cytoplasm contains the reticular network, which is formed by remnants of disintegrated organelles. Okay, formed by remnants of disintegrated organelles. Now, some organelles like the endoplasmic reticulum, they're degrading. Okay, organelles like the endoplasmic reticulum, the Golgi apparatus, they're degrading. So, because of this disintegrated organelles, it then forms a reticular network around the cell. So that's the major. That's why it gets its name, the reticulocyte. So that's the major characteristics of this cell. The reticulocyte is basophilic in nature due to the presence of remnants of disintegrated organelles. During this stage, this is the special thing that happens in this stage. During this stage, the cells enter the blood capillaries through capillary membrane from the site of production. Okay, during this stage, the cells enter the blood through capillary membrane from the site of production. This phenomenon is also known as diaperesis. Diaperesis, is a very important event during erythropoiesis. Okay, the red blood cells enter the blood capillaries through the capillary membrane from the site of production, the bone marrow. Okay, it moves from the bone marrow to the capillary, passes through the capillary membrane into the blood stream. Okay, so this phenomenon is known as diaperesis. Very important. Now, from reticulocyte stage, it now becomes the matured RBC. The reticular network disappears, and the cell becomes the matured red blood cell and attains the biconcave shape. The cell decreases in size to about 7.2 micrometers. The matured RBC is with hemoglobin but without nucleus, because the nucleus has disintegrated at the final phase of late normoblast. Okay, so let's look at an overview. We see stages of erythropoiesis, the important event that take place. In the PROERYTHROBLAST stage, is characterized by hemoglobin synthesis. Let's not forget hemoglobin synthesis. Early normoblast, the nucleoli begins to disappear. Intermediate normoblast, the hemoglobin starts appearing. Okay, the hemoglobin starts appearing at intermediate normoblast. Then late normoblast, the nucleus disappears. The nucleus disappears. Then we see the reticulocyte stage. There's the formation of reticular network, and then the cell enters the capillary from the production site. Okay, then the final stage with the matured red blood cell, the reticulum, as the reticular network disappears, and the cell attains biconcavity. Okay, so that's just stages of erythropoiesis analyzed simply for you guys. Okay, now let's look at the factors necessary for erythropoiesis. Without these things, erythropoiesis may not take place. Okay, we see that development and maturation of erythrocytes require various factors, which are classified into 3 categories. Now we have the general factors: erythropoietin, thyroxine, and vitamins. Let me just give an overview of erythropoietin. Erythropoietin is a glycoprotein, okay, with about 165 amino acids, and the major quantity of erythropoietin is secreted by peritubular capillaries of the kidney. Okay, a small quantity is also secreted from the liver and brain. Okay, what this erythropoietin does is that it stimulates the production of proerythroblasts from colony-forming erythrocytes of the bone marrow. Okay, it also induces the development of proerythroblasts into matured RBCs through the several stages. Okay, it also regulates, promotes the release of matured erythrocytes into the blood. Okay, the same thing thyroxine, the same thing vitamins. No, all is just geared into facilitating erythropoiesis. Okay, facilitating erythropoiesis, more erythropoiesis. Okay, so now let's look at the second category, which is the maturation factors. We have vitamin B12, we have the intrinsic factor of castor, we have folic acid. Okay, vitamin B12 is a maturation factor necessary for erythropoiesis. Okay, vitamin B12 is also called extrinsic factor because it is obtained mostly from diet. Okay, its absorption from intestine requires the presence of intrinsic factor of castor. So that's why most two of them are interchanged. That's why two of them are important. They're both maturation factors. So they help in the maturation of these red blood cells. That's from their name, maturation factor. They help in the maturation of these red blood cells. Okay, without vitamin B12, it causes what we call pernicious anemia. Okay, so because this vitamin B12 will not be absorbed from the intestine. Okay, it's not be absorbed from it's not be absorbed from the intestine, causing pernicious anemia. The red blood cells will not be matured properly, so it's anemia. Okay, also the deficiency of intrinsic factor of castor will cause vitamin B12 not to be absorbed from the intestine, which also causes pernicious anemia. So you see how both of them are very, very important. Folic acid also is very, very essential. Very, very essential. It is required for the synthesis of DNA. Okay, the synthesis of DNA decreases. This leads to anemia in which the cells are larger and appear in megaloblastic stage. Okay, without folic acid, the cells become larger and appear so megaloblastic, leading to an anemia called megaloblastic anemia. That's folic acid deficiency. So you see that these factors are very, very necessary for erythropoiesis to take place. Then we also see factors necessary for hemoglobin production. Well, you see such factors like iron, okay, which is necessary for the formation of the impact of hemoglobin. We see copper, which is also necessary for the absorption of iron from the GIT. We see vitamins like vitamin C, riboflavin, nicotinic acid, pyridoxine, so some proteins and some amino acids. Okay, so these are factors necessary for hemoglobin formation. So that will be the end of our video today. I hope you learned a lot from today's video about erythropoiesis. Okay, we looked at the stages of erythropoiesis. We look at the changes that happens in the cell due to the stages of erythropoiesis. We've also looked at the factors necessary for erythropoiesis. We have seen erythropoietin, thyroxine, vitamins, vitamin B12, intrinsic factor of castor. Okay, so I've learned a lot today. So I implore you guys to just like this video and subscribe to this channel to get more educational videos such as this on physiology and anatomy, and I'll be forever grateful. Okay, so thank you for listening. Goodbye.